Welcome everyone, please. Please take a seat. We will start on time. Thank you everyone for being there with us today. Thanks for, yes please Daniel. Okay, so after this quick general assembly, we have the pleasure to start our general meetings focused on clinical trials. Why did we choose this topic this year? Because we think that we are in a very, very important stage for our disease. We need your participation, your involvement as National Patient Association into this very important field, and it is a very important topic of clinical trials. We want to, during those 2 days, present to you everything on clinical trials, how to be involved, what are the stages, and of course for your patients, we need to be facilitator for patients to be included in clinical trials. The first session will be presented by Professor Divi Cornec, which I double thank because of his nightmare trip to come to Lisbon. So thank you so much, Professor Cornec, to being with us today. And he will start by presenting all RCTs in Europe. And you will see, but you obviously know, that we can have some hope for our untreated disease. And I take the opportunity to thank all of partners, because without this partnership, pharma companies, researchers, and patient association, it won't be at that level of involvement. So thank you all for being there. And Divi, the floor is yours. Thank you.
Thank you very much, Coralie, and a big thank to Sjogren's Europe for this kind invitation, even if the trip was indeed a bit tiring with a night at the airport, but I'm feeling good. So we will discuss about clinical trials, what is ongoing, but we will start with what we have done so far, what were the results of the different trials that have been organized in the last 3 decades, let's say, and what is ongoing right now. So I am from Brest in France, in Brittany, a very pretty region. Maybe one day we'll organize a Sjogren Europe meeting there.
We should have put that on the vote. New location, Brest, France.
So you all know that Sjogren's disease is a very heterogeneous disease. No 2 patients have the same symptoms and the same experience with the disease. The defining symptoms of the disease is this dryness of the eyes and the mouth that basically almost all patients feel. But some patients also have extraglandular involvement, what we call systemic involvement, and the disease can affect many different organs in the body. Another very critical symptom is fatigue, which is sometimes even more difficult to bear than the Zika symptoms. And so we started to stratify the disease based on those 2 facets, so the symptoms that all patients feel and the systemic involvement when you have inflammation in other organs. And so the clinical trials have been designed mostly based on those 2 facets of the disease. But we have a lot of recent biology data showing that beyond those symptoms, there is a high heterogeneity of the disease and with very different biological signatures between different patients. And so that may explain why the drugs so far have not proven their efficacy in many different clinical trials. And probably at some point in the near future, we will need to stratify the patients, meaning to give a drug to a given patient based on what we know of the biology of the disease in that patient. what we also know is that the quality of life when you have Sjogren's is deeply affected by the symptoms. When we compare, so we have some scores to kind of measure the quality of life in anybody, and so we use those scores to try to quantify the quality of life of patients living with Sjogren's, and we saw that the quality of life is really affected by the disease, even more than other autoimmune diseases that we follow in rheumatology and than other conditions. And what we studied some time ago is what explains this low quality of life. And the main thing that explains this quality of life is the symptoms of the patients themselves. And firstly, the fatigue and the dryness, much more than what we call the systemic involvement, when there is inflammation in other organs. So those two facets of the disease also have different impacts on the daily life of people living with Sjogren's. And so when we diagnose Sjogren's and we want to improve the condition, we can have two different objectives. First, we try to alleviate the symptoms, and that's all the symptomatic and replacement therapies. I will go quite quick on that part. And what we want to do next is to modify the course of the disease to try to have and to act on the root cause of the disease, which is the immunological system. You all know that Sjogren's disease is an autoimmune disease, meaning that the immune system creates itself the inflammation within the body. And so the drugs we have tested and the drugs we use and the drugs that are being developed all try to modify the immune system to decrease this level of inflammation. And so for the treatment of the symptoms, so ocular and mouth dryness, I will go quite quick because it's mostly replacement. So we lack tears, we give artificial tears. We lack saliva, we give artificial saliva, and a few other advice. But the few drugs that have been developed, so for example pilocarpine or cevimeline, only have a partial effect. And in my experience as a physician, it's not frequent that it really improves and for a long time the quality of life. So that's why most of the research effort currently is on the immune system and how can we modulate the system to improve the disease. We love drawing this kind of very complicated cartoons, but that is only to say that we have described in the last, let's say, 30 to 40 years which parts of the immune system are really important in the cause of the disease, in the what we call the pathophysiology, and at the center of the mechanisms leading to the disease, we have one type of cell called B cells. B cells are the cells of our immune system that make the antibodies. So they are very important, for example, for vaccination or to defend ourselves against microbes in general. They have many other roles, and when you develop an autoimmune condition, some of those B cells become kind of crazy and secrete antibodies that recognize parts of your own body. And so we really had tremendous improvement and new knowledge created those last 30 years about how those B cells function and how they are involved in the disease. And we mostly understood that they are not the single player, they are very important, but their interaction with the other cells of the immune system are also very, very important. And so most of the drugs that are being developed target some part of the function of B cells. Sometimes we want to target the immunoglobulins, so the antibodies themselves. Sometimes we want to kill the B cells, and we give drugs that destroy the B cells at least for some time. And sometimes we want to modulate them to try to calm them down because they are too activated. And so that's what we've done between 2000 and 2015. And very sadly, all those clinical trials have been negative. So I will just show a few examples. So that's another summary. For example, if we take the Joker trial, it was a study performed in France when we compared hydroxychloroquine, so Plaquenil, to placebo. When we want to prove that a drug has an efficacy, it's mandatory to organize what we call randomized controlled trials, and that's what will be the topic of those 2 days. So you will know much more about the design and how we perform those trials. But basically, the most important thing is to randomize the patients, meaning that it's only by chance, and it's a computer, that we allocate a patient either to the active treatment or to the placebo. And nobody knows during the whole duration of the trial whether a given patient got the placebo or the active treatment. And so we did that in France some years ago about hydroxychloroquine, because it was a drug very often used to treat Sjogren's. And so it was quite a large study with 120 patients, and half of them received placebo and half of them received hydroxychloroquine. And it's a perfectly negative trial, so you will probably see a lot of those this type of figures. So we observe along the time how the symptoms change, and you can see, and the black line is represents the patients who received Plaquenil, and the broken line represents the placebo, and you can see that there is no difference between the two lines and almost no change of the symptoms over the 6 months of this trial. There have been quite a few of these very negative trials. So for example, we also tested some drugs that work in rheumatoid arthritis, saying that the two diseases have some common points, and so we tested what we call anti-TNF. So infliximab was the first anti-TNF drug labeled for rheumatoid arthritis in 2000, so that was a long time ago, and definitely it had no efficacy in Sjogren's based on those symptoms. Then we tested other drugs, and we will speak a little bit about rituximab. You probably already heard about this drug. It's a drug that was developed initially to treat lymphoma because it targets B cells and it destroys B cells. And when we treat lymphoma, that's what we want to do. And we discovered a little bit by chance that it also improved autoimmune diseases. The first disease to be treated by rituximab was rheumatoid arthritis, and it was a big success. It's a drug that we use frequently, and so we decided to test it in Sjogren's too. And that's another large trial which was performed in France too, in different centers in France. And so we included 120 patients too. Half of them received rituximab, half of them received a placebo. So nobody during the trial knew who among the patients received either rituximab or placebo. And the definition of improvement in that trial was the improvement of the symptoms. So we wanted to improve the main symptoms of the disease, namely pain, dryness, fatigue, and the global evaluation of the disease by the patient. And unfortunately, this trial was completely negative also. 1/4 of the patients met the criteria for being considered as a responder in the rituximab arm and in the placebo, 23 versus 22% of the patients. So no difference at the group level 6 months after the treatment. But when we got a little bit more into the details of the symptoms one by one, we could observe some improvement. For example, on the bottom left, it's fatigue. So fatigue improved quite notably after rituximab, and dryness also improved. So there was some sign of effect or maybe efficacy of the drug on the main symptoms, but not at a level that we could detect using the definition of response that we decided to use in this clinical trial. And what was also striking is the effect of the drug in individual patients. You can see, so that's the evolution of dryness on the left in the patients who received placebo, and on the right who received rituximab. And we could see that some patients had really a strong improvement, like the dryness would go from 8 to 2, for example, in some patients, and in other patients there was absolutely no change over time. And that was even more the case with fatigue. So some patients really had a striking improvement of their fatigue after rituximab, and definitely more often in the rituximab group compared to the placebo group. So that led us to think that the way we defined the response to the therapy was not very good. So defining the improvement of those scales to assess fatigue, dryness, and pain. And so around 2010, we realized that most of the clinical trials were negative, and it was maybe partly due to the score that we used. The score we decided to use to define the response were definitely not sensitive to change over a period of 6 months, and we needed to create new tools to evaluate the disease. And that was done around that time, and we defined 2 new scores to assess the activity of the disease and the intensity of the symptoms. The first score is called ESPRI, and it's a score filled by the patient himself. So you tick the level of each of those 3 symptoms, and we compute the score as the mean of the 3 VAS. And more importantly, we developed the SDI, which is a score filled by the physician, by the doctor, to quantify the level of inflammation in organs outside of the glandular system. So to quantify the inflammation, for example, in the joints, in the lung, in the skin, in the nerves. And so this score is much more, the scale of this score is really broad. And so it created a hope that we could detect an improvement of the disease using this validated score. And it was validated in different groups of patients. And we showed that this score was probably a useful tool to do research. And so that's what happened after 2015. A lot of new clinical trials have been started, and all of them used the SDI as a primary endpoint to define what is the improvement of the disease. And luckily, fortunately, we had the first positive clinical trials in the last 5 to 6 years. So they are on the bottom of the picture. And those clinical trials used the SDI as a primary endpoint. But it didn't work for all the drugs we tested, of course, even if for each clinical trial there was a real rationale to test this drug. So some of those trials were negative. That's, for example, the results of the abatacept trial. Abatacept is also a drug labeled for rheumatoid arthritis, and we had some good scientific rationale to test it in Sjogren's, but unfortunately, we did not observe a strong efficacy of the drug. But we also had the first very positive trials. So this is the repurpose study that was performed in the Netherlands. And you can see that this time there is clearly a big difference between the placebo and the active arm. And in this trial, we evaluated not hydroxychloroquine alone, but the association of hydroxychloroquine plus leflunomide, which is another treatment of rheumatoid arthritis. And the association of the 2 drugs seemed to have a really strong effect on the SDI, so on the systemic involvement, meaning the inflammation in the other organs than the glands, but also on the ESPRI, so meaning the main symptoms, dryness, pain, and fatigue. And so this carried a lot of hope, and we are currently, I will show you later, evaluating in a much larger trial if this combination could be useful to treat the disease. And so a whole lot of new drugs have been invented those last years. As I told you earlier, most of them target B cells by some way. And so the first category of drugs is meant to kill B cells, to try to kill B cells with more efficacy than what we did with rituximab. And the drug that was the fastest in its development is called ianalumab. So it's a monoclonal antibody targeting a surface molecule on B cells, which is called BAFF receptor. And when the drug interacts with BAFF receptor, it depletes B cells, so it destroys B cells. And so there was two first small trial, and the first positive phase 2 trial. You will also discuss later in the seminar what are the different phases of the clinical trial design. So phase 1, phase 2, phase 3. But just really quickly, phase 1 is when you test a drug for the first time in a human. Phase 2 is when you test the drug for a disease in a small number of patients. And phase 3 is when you validate the efficacy of a drug in a large population of patients, very basically. So this was a phase 2 study, not so small, 190 patients, where we compare different doses of the drug. And you can see that on the SDI, there was a change in favor of the drug compared to placebo. And there was also another important result of the study, is that it improved the salivary flow. It could restore the function of the salivary glands, meaning that the patients at the beginning of the study had very low salivary flow, very low saliva in the mouth, and after only 6 months, we did observe an improvement of this salivary flow. And the effect on the symptoms in this study was not drastic based on the ESPRI, but there was strong results to be confident that this drug could be useful. And so the phase 3 trials were launched some years ago. Inclusion of the patients in those phase 3 trials was finished 6 months ago at spring. And so the last patient will have the last visit next spring. And so hopefully we should have the results of these Neptunus trials at next ACR meeting, so next fall in October, if everything goes smoothly. And it may be the first drug specifically developed for Sjogren's that would meet the criteria for efficacy in large clinical trials. Another way to modulate B cells is to change the way they get activated. And there are different pathways that lead to B cell activation. And one very important one is CD40 ligand system, meaning that when a B cell recognizes an antigen and wants to become activated, it needs co-signals. It needs to have other signals from other partners of the immune system. And one of these other signals is CD40 ligand. And it's involved in many different functions of the B cells. And so it has been hypothesized that modulating this CD40 ligand could dampen the overactivity of B cells and could improve the symptoms of autoimmune diseases. And so several drugs have been tested, and they are currently being developed. The first one, dazodalibep, is a small molecule that can interact with the CD40 ligand liaison, and that would change the activity of B cells. And the first clinical trial in Sjogren's was published just a few months ago. A bit larger than the previous trial. So 273 patients that received either this drug or a placebo, and in 2 different cohorts. A first cohort with patients included for systemic involvement, meaning with a high SDI score, and another cohort with mostly symptoms, with a high ESPRI and low SDI. And the study is positive, with a significant improvement of the SDI in the first cohort, and also an improvement of the ESPRI in the second cohort. So again, this is phase 2 results, so it's promising, but it doesn't prove the efficacy of the drug. And so the phase 3 trials for this drug also started a few months ago and are currently including patients. And here too, it seems that the drug could improve the salivary flow over time, so restore the function of the organs that are affected by the disease. There is another drug, this time it's a monoclonal antibody called iscalimab, that also blocks CD40, and so blocks the interaction between CD40 and CD40 ligand. Two phase 2 clinical trials have also been performed and showed a probable efficacy of this drug on the SDI, not so much on the S3, but it's a promising drug. For now, the development is on hold. There is no phase 3 trial organized yet, but it may come in the future. And lastly, there is a third drug targeting CD40 also, called abiprubart. It's also a monoclonal antibody that blocks CD40, and that will start or has just started a phase 2 study in Sjogren's that is for now running in the US, but will open in Europe early next year. And so all those three drugs target the same pathway, and so it probably means that this pathway is very important in the mechanisms leading to the disease. Next, we can try to change how the B cells are activated by blocking what happens inside the cell when it gets an activation signal. So in this time, we don't want to destroy B cells. We don't want to block something that happens outside of the cell, but we want to block something inside the cell, and that is called kinase inhibition. I will not go very far in the biology, but when a receptor is activated on the surface of a cell, it leads to a cascade of molecular events inside the cell, and we discovered that some of those events were really important to lead to the activation, and so some drugs were developed to block those activation events. And so the first drug, remibrutinib, blocks one of those kinases, which is called BTK, and there was a phase 2 study of remibrutinib in Sjogren's published earlier this year. You can see that all those publications are very recent, this year or last year. So it's very exciting. There are so many clinical trials ongoing. And there is also a signal of, for efficacy of the drug in Sjogren's, with an improvement of the SDI, an improvement of the salivary flow, and something to confirm on the symptoms of the patient. So positive signal, and for now this drug also is on hold. The phase 3 trial did not start, but it may come also in the future. And the other kinase that can be inhibited is called TYK2. It's a kinase that is important for the activation of the immune system in general. It's a drug that has been developed initially to treat psoriasis and other diseases of this family. And it has been tested then in lupus, and there was some efficacy in a phase 2 study in lupus. And so the development of this drug in Sjogren's started directly by a phase 3 study. And so the phase 3 study started earlier this year and is including patients based on the positive results in a very close disease, which is called lupus. And another way to modulate B cells, this time, is to get rid of the antibodies they secrete. I told you that one of the main functions of B cells is to secrete antibodies. When you have an autoimmune disease, you secrete autoantibodies, and so some drugs have been designed to get rid of the circulating antibodies. And so it's a bit more complicated to explain with simple words, but there is a system to recycle the immunoglobulins or the antibodies in our body, and mostly in the blood vessels. The antibodies can enter the cells, stay there for a while, and then get out of the cells and be active again. And so some strategies have been developed to block the system by which the antibodies enter the cells, and so they have to stay in the blood and they get eliminated much faster. And so this receptor is called FcRn because it's a receptor for immunoglobulins, And so when we block this receptor FcRn, we decrease drastically the level of the antibodies in the blood. And so those drugs were developed initially to treat some diseases in which we know that the antibodies themselves cause the disease. And the first disease that was evaluated is called myasthenia gravis. So it's a neurological disease that affects the muscles and the way the nerves can control the muscles. And in this disease, you have antibodies that block the communication between the nerves and the muscles. And so when we get rid of the autoantibodies, the disease improves. And so one of those drugs is called nipocalimab. And so it was tested in Sjogren's under the assumption that the autoantibodies or other antibodies in patients with Sjogren's are involved in the inflammation in the organs. And there was a phase 2 positive trial that was presented this year at our scientific conferences. And so the phase 3 trial for this drug is also starting and also including patients. And there is a cousin called efgartigimod, who is also blocking this FcRn system, and that is being tested in Sjogren's. So that's a lot of clinical trials, and all of them are currently recruiting with positive phase 2 studies, meaning that there is a real signal to consider that these drugs could improve the disease, and the first big phase 3 studies are being completed, and so we should have their results early next year. So if we meet again in 1 year from now, there will be some very important news because we will have the results of those first phase 3 trials. But it's probably not done, and we still have a lot of work to do to continue improving different facets. And the first thing we need to improve is the way we assess the disease. And that's the question that was envisioned for a big project in Europe called the Necessity Project. It's a project funded half by the European Commission and half by pharmaceutical companies to develop new tools to define what is the response of the disease because of a patient in a clinical trial. Because I see a lot of patients participating in clinical trials, and it's difficult to say, is it better? The best way to ask if a drug works in a patient is to ask, how do you feel? Do you feel better? And that's probably the simplest way, but it's not reproducible. If different doctors ask the same question to different patients, we may not be able to compare the results. So we need some validated tools, some scores, and that's the only way we will be able to show the real efficacy of drugs. And so in this project, what we did first is to get access to the individual results of all these clinical trials that were already performed. All of them were negative, but we had some clues to think that some drugs may have had an efficacy, and especially rituximab. Rituximab seemed to improve some of the features of the disease. And so based on all those data, we created a score that we called the STAR, the Sjogren's tool for assessing response. So it's a work that was performed and led mostly by Raphaël Serror, a colleague in Paris. And so we designed this score to better define what is the response to therapy. And so we try to take into consideration all the facets of the disease, meaning the symptoms of the patients, what we call the patient-reported outcomes, and the main score we have for now is the ESPRI, but also the evaluation by the physician of the activity of the disease, meaning the SDI, and the main features of the disease, which are the function of the salivary glands, the function of the lacrimal gland, so the quantity of saliva or its quality. For the salivary glands, we have a new tool to evaluate their morphology, it's ultrasound. I will just say a word about it. And for the eyes, there are also some specific tools that have been developed by the ophthalmologist to quantify the level of the disease on that part. And we also included in this system the main biological abnormalities, the lab tests that are abnormal in patients with Sjogren. And if those lab tests improve, it probably means that the disease improves. And so we tested this candidate response criteria in other trials, and we could have considered a posteriori that some of the trials could have been positive. So it's not allowed to do that in research, like to reanalyze a clinical trial. It's called post hoc analysis. So it does not prove that the drug works, but it gives really important clues to organize new trials. And so, for example, when we reanalyzed the rituximab trials, we could see a difference between placebo and rituximab using these criteria. So we would have needed to organize a new rituximab trial to prove that it works. But with the number of ongoing clinical trials that I showed you, it may have been a bit complicated. And so we decided to replicate the Dutch study, which evaluated hydroxychloroquine plus leflunomide. And in a big European context, so with different academic centers in Europe, we organized this clinical trial, which is mostly organized to validate the STAR response criteria. So it's the first trial that uses this criteria as the primary endpoint to define what a responder patient is. And so we are also still including the patients in this study. The inclusion will run until the first trimester next year, and then we will have 9 months of follow-up before the last visit. And so we should also have the results of this study probably early 2026. And so in this study, we included almost all the patients with Sjogren's, except those patients with a very low level of symptoms, because if the patient say, I have the disease, but my symptoms are very low, probably it's not a good idea to give him a drug if he's okay with the level of the symptoms. And so we include patients both with a systemic involvement and with mostly the Zika symptoms. What we also tried to do in this consortium is to develop new tools to evaluate the disease. And we developed a kind of diary, an electronic diary, that the patient can fill on a daily basis. Because the questions we ask to assess the level or the intensity of the symptoms, it's basically in the ESPRI. So the patient has to tick between 0 and 10 the level of intensity of the symptoms for dryness, pain, and fatigue. And we do that at inclusion, and we do that 6 months later, and we compare the numbers, and we say if it improves, it means that the disease improves. But what we know, discussing with patients, and it's the real life experience of most of the patients, is those symptoms are highly variable. From one day to the other, the dryness can change drastically. Sometimes it's okay, the dryness would be 2 or 3, and other days it would be 6 or 7. So comparing only 2 numbers is probably not a good idea, because at the beginning you could be on a good day and at the end on a bad day, and then we would say that the drug doesn't work, but maybe in between there was a really important improvement. So that's what we developed. So it's an electronic tool that the patient can install on his smartphone or tablet or computer, and we simply take the level of the symptoms on a daily basis. So during the duration of a clinical trial, the patient every day says, today my fatigue is this number, my dryness is this number, and my pain is this number. And what we did, we validated this tool first in a pilot study. We asked to 50 patients in my center in Brest and in another center in Paris to test the tool, just to install it in their normal life and to feel it for 3 months. And so what we observed is that first, the tool worked. That was a good thing. Second, the patients enjoyed using it, that it was easy to use and it entered their everyday routine, and it was not invasive, and it was positively viewed by all of the patients that tested it. And third, that the symptoms indeed are super variable. If you take, for example, the red line for this patient here, you can see that the symptoms go from 3 to 8 a few days apart. And so it's probably doesn't mean anything to compare this number with this number. It does not mean that the disease improved. It's just the normal change of the symptoms. And so now we are using this tool, the PEPS web app, within the Necessity clinical trial. So we had a big work to perform, and Coralie and the patients involved in Necessity were really, really helpful. We had to translate it into the different European languages and to adapt it to each of the systems in Europe. And we were able to do it. It's running, and so we will also have the results of this tool in a clinical trial, probably in 1 year from now, approximately. And last, another tool that we developed in the last 15 years, first to diagnose Sjogren's, is salivary gland ultrasonography. It's a very easy to use, oh, it disappeared, tool. Almost all rheumatologists have ultrasound machine in their room. And so we studied, and my colleague in Brest, Sandrine Jousse-Joulin, is one of the main physicians that led the study effort to use ultrasonography to diagnose disease. But we also wondered whether it could be useful to monitor disease, to see if we could observe some changes over time. And that would be really convincing, because it's kind of objective measurement of the disease. That we see abnormalities, if they go away, everybody will agree that it means that the disease improves. And so we tested it in our rituximab trial, so the TIERS trial, and indeed we observed, so on the left it's placebo, on the right it's rituximab, that almost no patient in the placebo arm had an improvement of the ultrasound compared to half of the patients in the rituximab arm. So probably this tool could also be useful to monitor the disease, and it is being used in different clinical trials. It goes with challenges because not all centers are experts, and so there is an important need to train the physicians to know how to scan the salivary glands using ultrasound, but it is being performed, and in the Necessity clinical trial we also included it, and almost all of the centers perform this study. And so if we look towards the future, another development, of course, is the progress we make in drug design and how we can make more powerful drugs. And one of the big excitation currently is the development of what we call CAR T cells. So what we want is get rid of the blue cell, the B cell, to improve the autoimmune diseases. And this is possible with a very powerful tools that are called CAR T cells, that were also developed to treat hematological diseases, so mostly lymphoma and leukemia. And the concept of those drugs is to take the cells of the patient himself, to transform them in a lab, to force those cells to recognize something and to destroy it. So we use the own cells of the patient as a weapon against something we want to get rid of. And so in cancer, we get rid of malignant B cells, so in lymphoma or in leukemia. And it was a real revolution for the treatment of those diseases. CAR T cells are much more, have a much greater efficacy than classical treatments such as rituximab, for example. And so it's being used, and it has been routinely used in hematology in the last 5 years, and most of the centers now are starting to use it. And since 2 years, the first use of those drugs to treat autoimmune diseases were published. And now there are, I just put a few, but there are so many clinical trials that are being organized to test those CAR T cells that could be the most powerful tool to get rid of B cells. And there are also, I will not get into details, but there are also other strategies that are being developed to better kill B cells, to try to get rid of them and to get rid of the autoimmunity. For example, what we call B-specific antibodies or T-cell engagers. And what we can also do is to organize mechanistic studies, because each time we organize a clinical trial, it brings us a ton of information on the study, on the drug itself, and on the disease. And so with Sjogren's, we have a really good opportunity to perform this type of studies, because the organ that is attacked by the autoimmunity is easily accessible. The salivary glands, we can perform biopsies. So it's the lip biopsies that are not very invasive. And most of the patients accept to have several biopsies so that we can observe what happens within the organs affected by the autoimmunity. This type of approach is not doable for many diseases. For other diseases, it's very difficult to perform biopsies. In Sjogren's, it's maybe not easy, but it's easier than in others, and it's acceptable by most patients. And so what we are organizing now, and we have several projects of this type, is to study how a drug works instead of if it works. Of course, if it's important to show its efficacy, but it's also very important to understand how it works and what is the global effect of the drug on the immune system. Because when we do that, we will be able to understand why the drug does not work in some patients, for example. And we'll be able to understand what signatures or what biological pathways are not targeted by some drugs. And so we are organizing this type of studies, and it goes with a tremendous improvement of the lab technologies. In our labs, we now have some really powerful tools to describe the immune system, both in the blood and in the tissues, in the biopsies. And so using those tools that generate mountains of data, it generates millions of data for each patient, we will really have a better understanding of the disease itself and how we can modulate the pathways leading to the symptoms. So to conclude, I'm on time? I didn't
Perfectly.
check.
Not like the planes, but you're on time.
So I hope that I showed you that the clinical research effort is quite recent in this disease compared to other diseases. It started basically 20 years ago with the first big clinical trials. The first ones were all negative, so it was a bit difficult to move forward, but we really developed new tools that changed the way we do research, and we study the efficacy of drugs. And we have witnessed a crazy acceleration of the research in this disease in the last 15 years. We at last have the first positive clinical trials, at last the first big phase 3 trials that hopefully will be able to prove definitely that some drugs can improve the disease. So we can be optimistic, and hope is permitted. And all of that, of course, would not be possible without the active involvement of the patients, because the doctors, the pharma companies work to create some tools to treat the patients, but the main and the central role in all of that is the patients themselves. So I think that what the foundations are doing all over the world, in the US, but here in Europe with Sjogren Europe, is really important. If we want to have drugs to be able to treat the patients, we need to get the patients involved themselves into those research programs. So thank you very much, and I will just show you how happy we are in Brest.
Thank you so much, Divi, for this very complete presentation. Before heading to questions, I would like to emphasize a couple of points. So yes, I think that hope is permitting, because at last Sjogren's get attention, and it moves very quickly. I hope you did catch that point. And as Davis said, if it moves so quickly, it's thanks to us, thanks to the pharma companies, and thanks to the researcher. So I think that this three stakeholders partnership is really important, and that's why we wanted to start with this presentation, again, because we, as patient association, we are facilitators for clinical research. In some trials in Europe, we know that they struggle to include patients. So it's also our role to promote research, to tell our members, our national, in your countries, where there are some clinical trials, to facilitate, to encourage patients to participate in trials. Because if we don't have any patients included in trials, we won't have enough results. So again, it's very important to highlight that particular point. We need to facilitate and promote research in our countries. Another point is that we haven't spoke about that earlier, but we have this year a collaboration with a medical writer, a journalist. And she will be able to deliver us some articles on these meetings. So that will be an important tool for you to communicate in your country with some articles ready to translate for you to your members. And another point, yes, the last one will be about the patient-reported outcomes. That's a great achievement, I guess, from, again, our collaboration with researchers and with pharma companies, because we all know that there's a huge difference, a huge discrepancy between our SDI score, let's say, and how we feel. And Divi has said it about the rituximab trial. And I think that a great success, and we can all be proud of it, is this new trial, including the patient's feeling. As you saw in many phase 3, there's one arm to assess the SDI, because of course it's important, but another arm, just like in necessity, who assess the patient's symptoms. And that's a huge step for us, because again, if we take rituximab, we all know that rituximab has worked for certain patients, but it has been declared negative. If the same trial goes today with our involvement as patient partners, with our pharma companies' partnership, maybe it would have been positive. So I think we need to go on that perspective of patient inclusion in research, and we will develop that, of course, these 2 days. Do you have any questions to Professor Kornek?
Thank you. Is this working? Yes.
Yeah.
So thank you so much for a very comprehensive presentation, very clear, very, how to say this, bringing us so much hope for the future. My 2 questions are, first on the PEPS web app, I understand it was developed for a particular trial, but do you think this can be used for clinical routine in the future? This is my first question. And the second question is about CAR T cells, of course. Do you think this will be used exclusively in refractory disease? Thank you.
Yeah, thank you very much. So for this e-diary that we developed, this PEPS web app, so it was developed initially in the context of necessity to try to be able to prove that a drug would improve the symptoms. But indeed, and that's what we've been discussing for a long time now, it could also be used as a personal diary, like to keep a track of how we feel, how the symptoms change over time. And that's actually what we did in our first pilot study. We gave the tool to 50 patients without any clinical trial or without any drug that we would change. And so they used it in their clinical routine for 3 months. And most of them actually kind of felt better because they were using it. So the main objective of this trial of this pilot study was to check that the tool worked, that it was usable, and so on. But most of the patients we proposed to use it were happy to use it actually, and wanted to continue for a longer time after those 3 months. And so within necessity, it's also the funding we got was from necessity, so from the European Commission for the clinical trial. But indeed, this tool now exists, and there is a real challenge to make it live longer, because it costs money, because you need some IT people to find the bugs and to make it run. And so that's something we started to discuss together, how we could invent a way that would give this tool to the patients, and that each patient at the individual level could decide whether he wants to use it or no. And it could also be used for patient-led research, the research led by the patients themselves. Like we created this tool, and it could be used by others, and that's exactly what we've been discussing with Coralie. It could be done by Sjogren Europe, giving access to this tool to the patients from the different countries, and it could be useful for themselves as dairy, and it could be also useful for research led by the patients. And that's a whole field of research that Sjogrens need to increase.
Yes, and we all know that when we monitor our symptoms, we can have, we can prepare the consultations with the doctors. We are not influenced by maybe a very bad week than we had, and we are convinced, and the research led by DV, we are convinced that it can be a very, very helpful tool for our members and for every single patient. So we will have the occasion to speak to you about this web app a bit later. We have a couple of, let's say, details to finalize, but it might be what Cathy has highlighted before, a huge, huge project for 2025 to offer this web app to all our members, of course, with the support of our partners, because as Divi said, it's a long development, and we need to achieve a lot of milestones before being able to deliver it to you at a European level. But it might be something that we will be able to discuss more precisely at the beginning of 2025.
And so for your second question, Anna, so for the CAR T cells, so they have been developed, as I told you, for leukemia and lymphoma, so a very serious disease with a risk of death, and so it really changed the evolution of those diseases. But even in hematology, they are used only for refractory diseases. They are never used as a first-line therapy, because it's quite the treatment itself goes with some risks, because you need to give a kind of chemotherapy before injecting it, and so it can have side effects. And then when you get rid of the B cells, it can also lead to other types of side effects. So we have to better understand the balance between the risk we have with this type of drug and the benefits we find. And so most of the time in this type of context, we offer this drug for the most severe cases, refractory cases, at least for the first, I don't say experiments, but just to get more experience with it. And then if we see that the tolerance is good, and that indeed it is spectacularly efficient for the most severe disease, then it will be evaluated in patients with milder diseases, and we will see. But for now, the format of this treatment of CAR T cells is a bit complicated, because you have to take the cells of the patient himself, send those cells somewhere for being engineered and modified, then they have to come back. So, and that takes at least 2 weeks. And so the patient has to stay in the hospital during that time, and then had to stay 2 more weeks in the hospital after we inject the CAR T cells. So it's a really heavy procedure for now. But there are some really huge developments to to try to develop CAR T cells that would not come from the patient, that would be there on a shelf and that we could inject to the patient at any time. So that would decrease a lot the complexity of the system. So it's called allogenic CAR T cells. And there have been the first reports of use of allogeneic CAR T cells for autoimmune diseases this year also from China, but it's very it's a few patients, like 3 patients, not Sjogren's but other autoimmune diseases. So it may come, but it's a bit early. And so it's very likely that the first patients that will receive the treatments will be only refractory patients before we have more data to potentially use these drugs for a broader population of patients.
And is this available, this kind of therapy? And this kind of therapy is available across Europe or just 2 or 3 countries? I'm thinking, imagine a patient in Bulgaria with a severe lung involvement and not responding to any other available therapy. Would this patient in Bulgaria
in hematology, it's becoming more and more used, and I guess in the different countries, it's a bit outside of my knowledge, but I think CAR T cells to treat lymphoma and leukemia are being used on a broader scale now. So for patients with autoimmune diseases, it could be, I guess, tried in different centers.
Can I ask something? I don't know if it's relevant or not to your question, but I realized that the recent last months, the recent year, that the patients asking about Sjogren, both by mails or in Greece by telephone and helplines, are increasing. That means that Sjogren's, by our awareness about Sjogren, is now recognized and diagnosed earlier, or it happens because some triggers from the environment make new, more Sjogren patients.
That's a tricky question.
Yeah.
That's something we evaluated in a very specific setting in the US, in the city of Rochester, in the county where the Mayo Clinic is. All the people living there accepted to have all their medical data used for research. And so we were able to evaluate the number of Sjogren's patients diagnosed every year over 40 years, between 1970 something and 2010 something. And so we did that some years ago, and we did observe a slight trend to increase in the number of cases diagnosed every year. But it also goes with what happened during that period. We changed the definition of the disease. Each time we create a new classification criteria system, automatically it changes the number of patients. And the second factor is the awareness of the disease. And that's your job, that's your role as a patient association to raise this awareness. Because, for example, when we speak with ophthalmologists, they see a lot of patients with dry eyes, and almost never do they wonder if it could be Sjogren's. And in my first plane yesterday, I was with a colleague ophthalmologist, and he told me, Sjogren's, it's rare, but it's mostly underdiagnosed. Like himself did not send the patients for evaluation. And so this is awareness, and this is really important, because it's obvious that when we diagnose a disease, most of the patients already had a long course of non-recognition of their symptoms. So that's a really important thing. So how we diagnose a disease, how people know about it and perform the diagnosis, so underdiagnosis. And then there could be also some environmental factors, but in my feeling, it's probably not very important for this disease. For other diseases, it's much more clear. For example, for inflammatory bowel disease, it's obvious that there is an increase. There are more cases now than there was 30 years ago. For Sjogren's, I think the main explanation of this slight increase we observed was the definition of the disease and the improvement of the underdiagnosis.
Any other question? Yeah.
I just have a remark. You just said when the studies need people, patients to be included, that's very good, but the SDI has to be quite high to get included. And there are loads of people just don't meet those criteria. So that's they would like to participate in some studies, but it's just not possible.
It's changing now. If you see the design, the protocols and the design of the phase 3 studies that Divis has talked about, I think, let's say starting with necessity, by example. Necessity, there's 2 arms. One for patients with SDI above 5, which is, Divi, correct me if I'm wrong, something roughly for 30% of Sjogren patients. And another arm with low SDI, but with Esprit above 5, which is probably here most of us, meaning 65%, 70% of patients. And that's, I would like to say that it's part of the result of patient association, speaking a lot with our pharmaceutical partners to explain that we want to be involved in clinical trials, but we can't because we are not as sick as the protocol study requires, an SDI above 5. And I really think that it's part of our work and it has been done because if you take the phase 3 clinical trial, just like the dazodalibep, by example, will include patients with low SDI and high Esprit just to explore if the treatment works on patient-reported symptoms. So I guess it's a change in the clinical research field, and it's not auto-congratulation, but I guess we are all part of this change by highlighting that particular unmet need about assessing our symptoms and just our symptoms. Thanks for us, we are not seriously impacted by the disease. I mean, SDI above 5, but we are really, really impaired by our symptoms. And that's the result of the collaborative work we are doing since now a lot of years with the pharma companies and, of course, the research team.